Right, so we are recording. So I'm going to go ahead and start our lecture for the nervous system. So we are going over exercises 12, 13, and 14. And I am actually going to pull up my PowerPoint for this. I know a lot of students have liked that so far this week. Okay, here we go.
Oh.
All right, so let me get to the beginning. Okay, perfect. So our nervous system, just in a nutshell, is going to be our command and control system for the entire body. So through our nervous system, we're able to gather just information in our environment, different types of stimuli, different things that are happening. We process that in our brain, and then our brain's going to generate some type of response to that. Okay, and a lot of times, you know, it's so subconscious that we may not even know that it's going on. And this is going to include things happening in the body. Okay, so we're going to be able to, our body is constantly gathering information on what's going on inside and then generating responses to that as well. All right, so we love to talk about our basic units of each system. So make sure that you know the basic unit of the nervous system is the neuron. Okay, again, that's in red. You know, things in red or in bold are going to be important for you to know. All right, so our neurons are going to be supported by our neuroglia or glial cells. All right, and they're going to have a lot of different functions, and we're going to talk about those in just a minute. All right, so this is going to be super important that you know these different terms and where they're going to be found on the neuron. Okay, so this is similar to our neuron model that I have the video posted of, and so this is a good picture for you to study. But the neuron model is what you'll see come up on the practical in terms of labeling and things like that. Okay, I will tell you there are two things labeled on here that you don't need to know. And so write that down, the telodendria and the collateral axons, okay? You don't have to know those, but everything else you do want to know. Okay, so I'm just gonna go through how a message is gonna travel through our neuron, okay? So when messages or electrical messages are traveling through our neurons, a lot of times they're going from neuron to neuron to neuron, and then they may end up at some type of endocrine gland or muscle cell, okay, lots of different things. So our message will be received by our dendrites, and then it's going to pass through the cell body, and then it's going to go through the axon hillock, which is this guy right here. It will then flow down the axon, through the axon terminals, and then to the synapse. We'll talk about the synapse in just a couple of slides. All right. So something else I want to point out to you are these little, they look like bubbles right here, but they're called myelin sheaths. And they're going to be made up of different types of neuroglia based on whether we're in the central nervous system or the peripheral nervous system. All right, so that's going to be important to pay attention to. And so the function of these is to kind of insulate that axon, and that's going to allow for messages to travel faster. Okay, because we want these messages, we want this information, these responses to be processed as quickly as possible throughout the body. All right, so like I said, we have different neuroglia in the central nervous system versus the peripheral nervous system. So the central nervous system is going to be the brain and the spinal cord, okay? The peripheral nervous system is going to be kind of everything else that we're thinking about. So any type of nerves that we see all throughout the body, those are all peripheral, and then our different nerves coming like off the spinal cord, those are going to be peripheral as well. All right, so let's look at our neuroglia of the central nervous system first. So first we have our astrocytes. This is just a cool picture. You don't have to know this. I'm not going to ask you to label it. We just have it here for fun. Okay, so we have our astrocytes, and where those are going to be located at, those are going to be around capillaries in the brain, in the spinal cord, okay? And so those are going to regulate what's going to come into the central nervous system, okay? Because in our capillaries, they allow for a lot of diffusion of different nutrients, different molecules, things like that, okay? So our astrocytes are going to be located there in those capillaries to allow for better nutrient exchange. Our microglia, these guys are phagocytic cells that destroy pathogens and debris. So what this means is that these guys like to eat up all the little things that they may see in the central nervous system that we don't need. Okay, so any type of cells that may have died, okay, anything that may have gone wrong in that process, okay, they're going to be eating those guys up so that they don't, so that these pathogens or these cellular debris do not stay in the central nervous system, because we don't want that. All right, so very important are oligodendrocytes. So our oligodendrocytes are the cells in the central nervous system that will be making up that myelin sheath that we see around the axon, okay? And then lastly, our ependymal cells are going to be producing cerebral spinal fluid. So our cerebral spinal fluid is flowing all throughout our brain in these structures we call ventricles that we'll talk about later on in this lecture. And so that's going to be circulating nutrients throughout the brain just to get to, you know, all of the different structures and things that we have. Okay. All right, so neuroglia of the peripheral nervous system. So both of these have similar functions to neuroglia in the central nervous system. So you want to make sure that when you're studying and going over this material, you really pay attention to those names, because literally every semester, every time I teach this class, students will get these confused, okay? I love to give y'all helpful hints just because I don't want y'all to mess up on little things like this, okay? So let's look at them. So our Schwann cells, those are going to be similar to our oligodendrocytes. So in our peripheral nervous system, they're going to be insulating that axon that is, they're going to be insulating the axon. They're going to be making up the myelin sheath. Okay. And then our satellite cells, those are going to be kind of like our astrocytes. So they're going to be around the body of the neuron, kind of regulating what can come in and out. Okay. Again, you don't have to know this picture. It's just for fun. All right, so this is going to be a super important slide, y'all. You want to know this figure. You want to know everything that's labeled on it. And then you also just want to know kind of this process of what is going on right here. So I'm going to go over this, but just make sure that you are kind of paying attention. All right, because I have my PowerPoint up, I can't see any questions that y'all may have if things are coming up. So I'm not ignoring you, I promise. All right, so when I get to the end, I'll take a look at any questions that may have come up and we can go back to those, okay? So like I said, this is our synapse, okay? And so we have right here the presynaptic membrane, we have the synapse itself, and then we have the postsynaptic membrane. So our presynaptic membrane is going to be our axon terminal, really, okay? And so see these little purple bubbles right in here? Those are going to be our, those are going to be called synaptic vesicles, and they're going to be filled with neurotransmitters. Okay, and so we see right here as our neurotransmitters in our synaptic vesicles are flowing through the axon, and they come right here to the synapse, and then they are released. Okay, so those vesicles open up, releasing that neurotransmitter into the synapse. All right, and then those neurotransmitters are going to bind to receptors on the postsynaptic membrane. All right, and so that postsynaptic membrane, like I kind of mentioned earlier, can be a couple of different things. It can be a dendrite of another neuron. It could be an endocrine gland. It could be a muscle cell. Okay, so it could be a lot of different things. All right, so it'll bind to that receptor and then continue the process. Okay, so super important slide. Actually, I'm going to run back to our post right here. Okay, just questions. Let's see. If you watch the recording later, when is the discussion post due? By Sunday night, just when everything else is due. Practical will be in 3 weeks. Okay, and don't worry, you won't miss it. I'll make sure that I let y'all know when everything is. Okay. All right, so back to our PowerPoint.
Financial aid.
Okay, so if you remember a few weeks ago, you know, before spring break, when we were on the muscular system, we talked about some of that organization. So hopefully you kind of remember kind of those levels and maybe some of those words, because we see them here again, okay? So we see the prefix endo, we see peri, and we see epi, okay? And so endo, that's kind of meaning, you know, like within, peri is around, and epi, that's going to mean above. All right. And so just kind of go with me as we're going through this figure, which you will want to know. Right. So we start with our axon right here, covered by the myelin sheath, and then that axon is covered by the endoneurium. Okay, so axon, myelin sheath, endoneurium. Okay. Okay. Guys, I'll get to practical questions at the end. I see them, but I'll get to them at the end. Okay. So then we have our perineurium. Okay. And so in the perineurium, we're going to have a lot of, we're going to have all these different axons covered in their myelin sheath, covered in their endoneurium. We're going to have a bunch of those together. That's going to make a fascicle, which is going to be covered by our perineurium. So you can see that right here. Okay, we see our fascicle, we see our perineurium. All right, and then we're going to see a bunch of fascicles kind of bundled all together, as well as some blood vessels, all bundled together, and that's going to be covered with the epineurium, which is going to make that entire nerve. Okay. No. So you definitely want to understand those terms, or you want to understand this in the terms, but as well on the figure. Okay, so talking about brain regions, we're kind of switching gears just a little bit. All right, so we're going to say that there are 6 brain regions. So some textbooks might say that there are 4 because they'll consider the brainstem to be one whole region. Okay, but we're going to say that there are 6. All right, so we're going to talk about each of these just kind of basically. All right, and for the next few minutes, I'm going to be talking about some information that is not on the PowerPoint. It's not on your study guide, but you still want to know it. So it's kind of my incentive to get everyone to watch the video, okay? Because this is extra material that you will, be responsible for. All right. So in our cerebrum, all right, so that's our big main part of the brain. We see that there are these 4 different lobes. So we see our frontal lobe, our parietal lobe, our temporal lobe, and our occipital lobe. So if you remember from our few weeks when we talked about the skeletal system, we learned the bones of the skull, okay, and they're in those same locations. So hopefully this won't be new to you. You'll be able to learn those really quickly, okay? We also see that our cerebrum is kind of split in half into 2 different hemispheres, okay, left and right, all right? And we'll talk a little bit more about that in just a minute. So I'm going to talk about each of these lobes individually, talk about their functions, and then talk about some problems we can see within them, okay? So first, our frontal lobe. So this guy is going to be super important because we're thinking for our frontal lobe, that's going to really be where any type of executive function is going to be happening. And so when I say executive function, I mean like decision making. So you made the decision to jump onto Zoom for this lecture today. All right, so that was your frontal lobe. All right, next we have our parietal lobe. So that's going to be like directing movements toward some type of goal. So like you want to do some type of task. So I want to pick up my pen and I did the movements necessary to pick this pen up. So that's going to be my parietal lobe at work. All right, so next we have our temporal lobe, and that's going to be involved with hearing, language, and musical ability. So you hearing me right now, that's your temporal lobe at work. And lastly, your occipital lobe is going to be visual processing. So you seeing me, you seeing my PowerPoint that's up, that's your occipital lobe, okay? All right, so I want to talk about 2 specific areas that we see within different lobes. We have Broca's area and Wernicke's area. Okay, so I'm actually gonna like type those out for you so you can know how to spell them. Okay. So we have Broca's area and Wernicke's area, okay? So Broca's area is gonna be in our frontal lobe, okay? So right up here in our frontal lobe. And so this lobe is really important because it helps with the motor components of speech, okay? So in our frontal lobe, it helps with, moving our mouth, using our tongue to create speech and to produce that. Okay, so if we have a problem here, that's going to be called Broca's aphasia. Okay, so I'll write that out for you. Okay, Broca's aphasia, and then we also have Wernicke's aphasia. Okay, so with Broca's aphasia, we have some type of problem in producing speech. Okay, so a patient who's going to have Broca's aphasia, they can understand the speech of others. So someone with Broca's aphasia could understand what I'm saying right now, but could not verbally answer a question or repeat anything that I'm saying. Okay, they can understand it, but they can't produce that speech in response. All right, and so for Wernicke's aphasia, again, we have some type of problem with what's going on there. So in Wernicke's area, we're going to see that that's going to be in both the parietal and the temporal lobe. All right, and so this is going to be sensory. So Broca's is motor, Wernicke's is sensory. So in Wernicke's area, that's going to help you to understand speech and allow you to kind of recognize and understand the right words for that thought, okay? And so for a patient with Wernicke's aphasia, they can produce speech, okay? So they can form those thoughts, they can produce that speech, but they cannot understand what others are saying, okay? They can't understand the speech of others, all right? So it'll be super important that you know just both of those terms, where they're located, what they do, and kind of those consequences of issues there. All right, so next, this is also not on your slides or on your study guide, but we're going to talk about the different types of stroke and kind of what happens there, what symptoms of strokes are, things like that. So a stroke, just in a basic definition, is going to be some severe interruption of blood flow in the brain, okay? So we have two different types. We have ischemic stroke and we have hemorrhagic stroke. So in an ischemic stroke, that is when there is a blood clot in some type of blood vessel in the brain that's blocking blood flow. Okay, so that's a blood clot. Then we have a hemorrhagic stroke, and so in a hemorrhagic stroke, some blood vessel in the brain has burst and there is just severe uncontrolled bleeding. But as a result of that burst and that bleeding, some area of the brain is not getting the blood that it needs to function, okay? So a lot of problems there. So our main symptoms of a stroke are gonna be facial droop. So it's gonna be one side of the face that's gonna be drooping. All right, motor weakness in the limbs, visual impairment, headache, and speech difficulties, okay? So those are a lot of the telltale signs of a stroke, all right? We really don't see a lot of treatments for that stroke itself. A lot of times when we're thinking about treating a patient who's had a stroke, it's really more treating what has happened as a result of that stroke. Out of every 10 people who have a stroke, 2 of those people die, and then of the rest of those people, most will have some type of cognitive deficit. So there's going to be some type of irreparable damage in the brain as a result of that interruption of blood flow, okay? However, for our ischemic stroke, we do see a treatment that can really help patients who have this type of stroke. However, a lot of times it's too late to use it by the time that the patient knows they are having a stroke and can get to the hospital to get medical help. All right, but that treatment, you want to know this, is called tissue plasminogen activator. All right, I'll write that out for you. Tissue plasminogen activator, also abbreviated as TPA. All right. And so this functions to break up that blood clot in whatever blood vessel we see the stroke in. So it's going to break up that blood clot and allow that blood to continue to flow. And it can actually help a patient by a substantial amount. However, that's if it can be used in time. All right. Okay, so kind of moving on just a little bit. You want to know some of these structures that we see, you know, and kind of what the cerebrum looks like itself. All right, so we see this thing called a gyrus and then this thing called a sulcus. All right, you can also see these when you're looking at the brain model that I have a video of. So our gyrus is going to be like a fold. It's going to be like the little flat surface that you can see on the brain. And then a sulcus is going to be the little valley in between each gyrus. So in between each of those little folds, okay? We also have this structure called the corpus callosum. We can see it right here, but you can see it much better on the video of the brain model that I posted. And so the corpus callosum is going to serve to connect the left and the right hemispheres, and it's going to allow for them to send signals between each other, which is going to help for us to have coordinated movements. All right, so I can raise both of my hands at the same time because of my corpus callosum. All right, so it allows both sides of the brain to really work together. And you also want to know that the corpus callosum is made up of white matter, okay? And we're going to talk about the difference between white and gray matter in just a minute. Okay, so moving on to the next region of the brain. So we have the cerebellum. So the cerebellum is located on the posterior side of the brain, kind of inferior to the cerebrum and kind of on the brainstem and the spinal cord, okay? So it is also divided into two different hemispheres, okay, left and right. And we see this unique structure. It's really cool looking, actually, called the arbor vitae. And it's these little white projections that you can see right in here that kind of flow throughout the whole cerebellum. Okay, you can see it really well actually on the sheep brain dissection video that I have posted as well. All right, and so the cerebellum is going to be really important in functioning to monitor body position and coordinating our movements. All right, so someone who may, if someone falls and they hit the back of their head, they may not be able to walk straight. They may have some problems with their coordination, and so that's damage to the cerebellum. All right. Okay, so for our next region, we have the diencephalon or the forebrain, and that's going to be the thalamus and the hypothalamus together. All right, so right here, this is our thalamus, and then this right here is our hypothalamus. And so if you remember what the prefix hypo means, hypo is going to mean below. So if you find the thalamus and you look directly below it, that's where the hypothalamus will be. All right, and so our thalamus, that's going to be working to interpret lots of different types of sensory information, and our hypothalamus is going to be linked to the endocrine system, but also works with our emotions and instincts, okay? And I know these seem like really broad, really vague definitions, and that's because they are, okay? There is so much more to both of these parts of the brain than what I'm listing, but this is just all we want you to know. We just want to keep it simple for lab. Okay, so moving to the brainstem, we have three different regions: the mesencephalon or the midbrain. You can say either one, that's fine with me. The pons and the medulla, okay? Or medulla oblongata, you can say either one. And so altogether, the brainstem does some basic survival tasks, housekeeping things, okay? Which you can kind of see by the little bullet points that I have on here. Okay, so our midbrain or mesencephalon, that's going to be for processing different visual and auditory stimuli and reflexes that we are taking in subconsciously. okay? Next, the pons is just going to be a relay station for sensory information, and the medulla is going to be for coordinating our swallowing reflex as well as heart rate and breathing patterns. Okay, so very, very important. All right, and so on the model, on the brain model, I point all of these out individually. And then on the sheep brain dissection, guys, our sheep brains are a little sad because in the packaging, they get kind of smushed together. So sometimes it's really hard to see some of the structures. However, if you can kind of remember the order of these different regions of the brainstem from superior to inferior, then you'll be fine. And if I have something tagged on the sheep brain that's one of these, I'll do my best to make it very clear, or I might even like draw little divisions out so that you can see it, okay? So if you remember that the mesencephalon is the most superior, then you have the pons, and then you have the medulla, then you're gonna be fine. All right, so as promised, moving to our ventricles. All right, this will be a really important picture for you to know. This will be on your practical. Okay, so you definitely want to know these different parts. Okay, I'm not going to go over all of them individually, but basically all of our ventricles, all of these different structures, they're all working together, they're all connected, and we have cerebral spinal fluid that's going to be produced within them and circulated throughout. So most of our cerebral spinal fluid is going to be made by our ependymal cells. So remember, central nervous system, ependymal cells make cerebral spinal fluid, and they're going to be in the choroid plexus. So the choroid plexus isn't really shown on here, but it's kind of around this area if you can see where I'm moving my mouse. Okay, and so the choroid plexus is like this, just big mass of capillaries in the third and the fourth ventricles. Okay, so moving on. So next we have these things called meninges in the brain. And so these are going to just be layers of tissue that are laying on top of our brain. So you want to know them from superficial to deep. You want to know the names, all right? And then anything else that I have mentioned right here. So most superficial, we have our dura mater, which is going to have two sublayers, our periosteal layer and our meningeal layer. All right, below that we have our arachnoid mater, and then right below the arachnoid mater we have what's called the subarachnoid space, and we're going to see a lot of cerebral spinal fluid in there as well. And then just deep to that we have our pia mater, which is a very thin layer. It's much thinner than the other layers. Okay, we have the pia mater. You also want to know that when any of these meninges are infected, all right, or inflamed would be another word that we could use, all right, that's going to be meningitis. Okay, let me color that red. So meningitis, very important for you to know. Also, just a little tip, if you ever see something that ends in itis, so I-T-I-S, it ends in itis, that means that there's some type of inflammation going on. Okay, just a little side note for your reference. All right, so our cranial nerves, all right? So usually students hate this in labs because you do have to know the, I'm looking through the lab manual to find something right now, but you do have to know each of the nerves, whether they have a sensory function, motor function, or they function as both, and their actual function as well. Okay, so in your lab manual, you have this awesome little chart right here where it tells you all that information, really good stuff, okay? And normally on your practical, you would just get a whole entire blank chart and you'd have to fill everything in, okay? And that would be a chunk of points. However, we can't really do that for the practical. So the way that I'll be asking you questions about this is I'll say, for example, does the facial nerve have sensory function, motor function, or both? And so you would tell me both. Or if I said that a patient has suspected nerve damage, their symptoms are they're having problems with hearing and balance, what nerve may be affected? You would say cranial nerve number 8, vestibulocochlear. Okay, so those are the type of questions that I would ask you, but this chart is super, super important. You definitely need to know it and definitely need to bookmark that in your lab book. However, I know some students don't have their lab manuals because you're home right now and your lab manual may be back at school. And so if that's the case, if you are missing homework because of that, or if you need this chart, anything like that, you could just let me know, just like shoot me an email and I'll send that to you, no problem, okay? All right, so I'm not gonna go over all of this individually. Like I said, I'm just gonna leave that to you to study on your own. Okay, so the next few slides are all chapter 14. Okay, I'll get my notes. All right, so all chapter 14. All right, so we're looking at the spinal cord and we see that there are 4 different regions. All right, these are the same as the names of the vertebrae that we learned about a few weeks ago. So we have cervical, thoracic, lumbar, and sacral. Okay, you definitely want to know that there are 31 pairs of spinal nerves that come off the spinal cord, and those are all represented right here in this figure. You don't have to know this figure. This is just for you to kind of get a visual of what this looks like, okay? Let's see. Okay, so next I want you to know the difference between gray matter and white matter. Okay, so we see this in the brain and the spinal cord, so in the central nervous system, all right? And gray matter, that's gonna be what we see or where we see our neuron cell bodies and our unmyelinated processes from the neuron. And then in our white matter, that's where we see all of those myelinated axons, okay? And so you definitely just want to know the difference between those. Normally, I would give you a model that looks kind of like this figure and then have things labeled, but I'm not going to make you know that model just because of all the change and transition going on right now. So just know the difference between gray and white matter. All right, next we have dermatomes. And so dermatomes, those are gonna be different zones in the body that each spinal nerve will serve. Okay, so we have a cervical zone of dermatomes, we have thoracic, lumbar, and sacral. And those are all shown on this guy right here. And so I'm not going to ask you to like list like what I have in bullet points out here. I'm not going to say, tell me where the cervical dermatomes are. I'm not going to do that. I'm going to have a figure up like this, except it's not going to have the letters and numbers on it. And I'm just going to point to a region and ask you what it is. Okay, pretty basic. All right, so you just want to know those names and just be able to locate that region on a figure like this. Okay, guys, we're almost done. So normally I would have this whole figure. So on the practical, normally what I would do is I would have this whole figure on there and have a bunch of things kind of blanked out, and you'd have to like fill in the blank. I think last semester I had like 7 questions from it, okay? And actually students did really well because I remember I hated having to learn this when I took anatomy. Okay, so this picture up here with the little guy, this is just for you to kind of see what it looks like, where it would be. All right, this is the main one. And so I haven't decided how I'm gonna ask for this on the practical. I could still have things blanked out and ask you to label, or I could say there was nerve damage at there was damage at some specific place, what major terminal branch would be affected, okay? Or what major terminal branches could be affected, because that could be multiple ones, okay? So I would just recommend going over this and like being familiar with like these different roots and how those kind of converge to form trunks. And then we see them kind of diverge into these divisions, and then they come back together for cords, and then they break off into these terminal branches, okay? So just kind of go through that, kind of look at those pathways, all right? And just kind of just go over that figure and you'll be fine, okay? So next, action potentials and neurophysiology. So again, in your book, you have this worksheet. I'm not going to ask you to like turn it in for your homework, okay? But you can read a little bit about action potentials here. And then on the back side right here, you can read a little bit more. There's an awesome figure and then some questions, okay? I definitely recommend that you still go over those questions and read through this because this will be on your test. And I'm actually not gonna go over it in this lecture today because I have a recorded version of that where I kind of draw it out and I go over all the terms individually. And just because of this setup, I really can't do that here with y'all. So I'm gonna post that when I post the recorded lecture. Okay, so make sure that y'all watch it so that you can kind of see me go through all of that and kind of tell you what you need to know. All right, and then lastly, just a couple things on the sheep brain dissection. A couple things I'm gonna tell you that you don't have to know. You don't have to locate the optic nerve or the pineal gland, okay? Optic nerve and pineal gland, you don't have to locate those, but everything else on here is going to be fair game, okay? And I go over, again, everything that you need to know on your, on the video, on the sheep brain dissection video that I posted. Okay, so that is the end of the PowerPoint. So I'm going to go back to our regular screen now and answer any questions, but that is the end of the lecture. So I'm going to end the recording here for y'all.